Okay, so I'll be telling about in today's lecture how in the immunology how that there are set of B cell and T cells. So in the last lecture we read about the T cell developments, and in today's lecture we will be studying about B cell development. So how the B cell develop, where it develops, what is the site of origin, what is the site of maturation, what is the site of activation, and what are the different processes through which it gets activated and becomes a mature B lymphocytes. Okay, so the origin place is the bone marrow for our lymphocytes. So in this bone marrow, so the bone marrow is having the multipotent progenitor cells. Okay, from multipotent progenitor cells we get the lymphoid progenitor cells. So these are firstly hematopoietic stem cells make the multipotent progenitor cells, which produce lymphoid progenitor cells. So from this lineage we'll get three kinds of cell. One is pre-natural killer cells, pro-B cells, and thymocytes. Okay, so pre-natural killer cells gets mature and make the natural killer cells, which help in the antigen presentation and killing of the pathogen. And then other variant we get is the thymocyte, which develops into the mature T lymphocytes. Okay, and in this T lymphocytes we get T helper cells and T cytotoxic T cells, and here we get the pro B cells. So in this B cell it gets uh, mature. So from B cells we get the pro B cells, we get the B cells, and then they get mature. So then these mature B cells go into the periphery to the lymph node, and there when they experience the antigen, with the antigen interaction, antigen and this B cell interaction, it start proliferating, and with the help of T helper cells it start proliferating and divided into two types of cells. One is the plasma cells, which secretes the antibody. Another is the memory B cell, which acts as a memory cell, so that when the next infection comes they will immediately start their effect in a very faster manner. Okay, so if we'll start from the very beginning, so initially these start with a multipotent progenitor cells. Okay, as I have mentioned. So this multipotent progenitor cells, these are having a receptor on their surface, a surface receptor which is called as FLT3. Okay, and there are stromal cells present in this bone marrow in the surrounding, and these stromal cells having the ligand against the receptor. So this ligand is FLT3 ligand. Okay, so this is for this multipotent progenitor cells, which expresses FLT3 on its surface, gets attached to the stromal cells via the ligand FLT3 ligand. Okay, now next what happens due to this interaction, now it matures into the lymphoid progenitor cells, and when it matures into the lymphoid progenitor cells and start expressing the IL-7 receptor. Okay, IL-7 is secreted by the stromal cells. Okay, so IL-7 is secreted and attached to the lymphoid progenitor, the receptor present on the lymphoid progenitor cells, which is IL-7 receptor. Okay, from there these cells converts into the pro B cells. Okay, so the progenitor B cells, these progenitor B cells now start expressing this particular surface marker, which is called as BLA4. Okay, BLA4, and another is c-KIT. Okay, BLA4 and c-KIT. So BLA4 is a kind of integrin, which adhesive molecule expressing on the surface, which adheres with the stromal cell with the help of VCAM-I. Okay, and here another interaction which is happening is c-KIT, which stem cell factor interacts with the stem cell factor present on the stromal cell surface, and also ILR7 receptor is there, which interacts with the IL interleukin 7, which is secreted by this stromal cell. Okay, so all these interactions are happening. So other than that, this in the very beginning, this tyrosine kinase receptor, okay, so it with this FLT3, it is a tyrosine kinase receptor, and there is this one particular chemokine which is CXCL12. So this particular chemokine tries to adhere the multipotent progenitor cells with the stromal cells, okay. So it helps to retain these multipotent progenitor cells to stay on the stromal cells. So these are the very important chemokine CXCL12, which tries to retain or the MPP, the multipotent progenitor cells, by adhering it to the stromal cells, which converts into the lymphoid progenitor cells, start expressing IL-7 receptor, which converts to the pro B cell, which expresses VLA4 integrin and c-KIT on its surface. VLA4 interacts with the VCAM1, and this is stem cell factor interacts with the c-KIT. Okay, then it is called as progenitor B cell. So after that, it gets converted to the pre-B cell. Okay, the pre-B cell. So now this pre-B cell start expressing the antibody on its surface, the IgM on its surface, okay, along with Ig alpha and Ig beta as a receptors, okay. And then also then this IL-7, it plays a very important role. So this particular IL-7, as I have already mentioned, stromal cell express or secrete this IL-7 interleukin 7. So again, the interaction of IL-7 to this pre-B cell leads to detachment of B cell from the stromal cells, okay. And then this pre-B cell matures into the converted into the immature B cells. So this immature B cells is expressing the IgM on its surface along with Ig alpha and Ig beta. This immature B cells, when it starts expressing the IgM and IgD both antibodies on its surface, then it is called a mature B cells. Now this mature B cells, it goes to the lymphoid tissue in the peripheral system, so that it can encounter the antigens now, okay. So this is what the maturation through which the B cell go through, okay. So the B cell starts with the multipotent progenitor cells, which expresses FLT3 tyrosine kinase receptor, interacts with the FLT3L. So there this chemokine CXCL12, which tries to retain the multipotent P cell in the here and try to keep it attached with the stromal cell. Then this lymphoid progenitor cell start expressing IL-7 receptor, which interacts with the interleukin 7 and is secreted by this stromal cells, which gets converted to the pro B cells. Now this pro B cells have the integrins VLA4 and the c-KIT. VLA4 interacts with the VCAM1 and this c-KIT, which is also a tyrosine kinase receptor, and interacts with the stem cell factor present on the stromal cells, gets converted to the pre-B cell. These pre-B cells now start expressing the IgM immunoglobulin molecule on its surface, and then it converts into the immature B cell. And then immature B cell, when it starts expressing both IgM and IgD on its surface, then it is called mature B cell. Then so through that maturation process, this is a more detailed view, okay. So from the stem cell, when it starts, so in the pro B cell, here at this pro B cell also, it start expressing. So in this pro B cell also, there are two steps. There is a early phase and there is a late phase in this pro B cell, okay. So what happens there? So as we know that these B lymphocytes, as a surface receptor, they are having antibody molecules on them, okay. So those are those surface receptors of B lymphocytes. So in the previous classes also we have read about the when in the antibody molecule, there is a heavy chain, there is a light chain, and when we start reading about that, we read that there is a recombination events, and there is a V chain, there is a D chain, there is a J chain, which is there, which is present in the antibody molecules. So there are recombinations happen. So there is a first in the from heavy chain and light chain, the heavy chain recombination starts. So there you have this D region in the heavy chain only, so which brings the diversity into the heavy chain portion. Okay, and so and due to that, it can interact with several kinds of antigen. Okay, so that D region is present. So when antibody molecules start synthesizing, okay, so that time this recombination events happen. So there is V, D and J region. Okay, so when there is these regions are present, the first recombination takes place between D region and J region. Okay, and then after DJ they join with each other, then they join with the V chain. Okay, so then VDJ recombination occurs. Okay, and then the light chain rearrangement occurs. So in the light chain we do not have the D region, we have only V region and J region in the light chain. Okay, so here if we'll see in the pro B cell, okay, it can be divided into the early pro B cell and late pro B cells. So here in the early pro B cell, you start the in the starting of this when the stem cell converts into the pro B cell, in the early phase the DJ recombination occurs. Okay, this H in the J denotes here that this is here we are talking about the heavy chain. So there the D region and J region, they recombine with each other. So because as I mentioned in the light chain also this rearrangements occurs, but there is no D region for the diversity. So whenever we write the V or J, so it is for, if it is for both, to differentiate, so for V we write this VH or JH of the heavy chain, or VL or JL for the light chain. Okay, so this is the rearrangement process starts. So first in the early phase the DJ recombination takes place, followed by the late phase. So in the late phase, after the DJ recombination, VDJ joining takes place. Okay, DJ joining followed by VDJ joining during the late pro B cell, and there only we start having this Ig alpha and Ig beta arrangements. So then now this in this heavy chain the rearrangements are done. Okay, so this VDJ is rearranged. After that it goes to the surface, and then it's now you can see this blue chain, it is the heavy chain of the antibody, and this is Ig alpha and Ig beta. Okay, now in the red one as a light chain you can see, so they take a surrogate light chain. Okay, so in the surrogate light chain they take up this sequence V pre B and lambda 5. So this is the surrogate light chain sequence. Okay, so this is the surrogate light chain sequence which adapts here, which, and when the cells start expressing this heavy chain, a fully VDJ rearranged heavy chain along with the surrogate light chain and Ig alpha and Ig beta, at this stage we call it as pre B cell. Then it gets then when the light chain rearrangement is complete and the surrogate chain is replaced by the original light chain, then it becomes a then at this stage we call it as immature B cell. From pre B cell we call it to immature B cell, and it expresses IgM on its surface. Then when the cell starts expressing both IgM and IgD on its surface, it is called a mature B cells. Okay, so early phase DJ rearrangement, late pro B cell VDJ rearrangement, pre B cell we have VDJ rearranged heavy chain along with surrogate light chain, and then in the immature B cell the IgM expressing, mature B cell IgM and IgD both expressing on the B cells. Then they go into the circulation and in the lymph node where they encounter the antigens. Okay, so here you can see this is on the surface. So first thing is it interacts with this antigen presented on the other cell surface. Okay, and so for example here a cell is presenting antigen on its surface. Okay, so here you can see these are the immature the, the mature B cells. It is interacting with the antigen, and yeah, so then here also is when the mature B cell goes to the periphery in the circulation. So this is a quality test or quality control check test. Okay, so this is the test. So when the mature cell starts exiting, before that they encounter the antigen, self antigens. Okay, so if um, these cells, it does not encounter the self antigen, because here in this process you have seen a lot of VDJ rearrangement has happened, and due to those rearrangement, the cell has or the B cell has acquired such affinity that it can interact with its own antigen and try to kill our own cell. So those B cells should not be selected. So that is why this quality control check start. And here in this process, if it can interact with the self antigen, it will not be selected. So if here it does not interact with the self antigen, then it will be passed and goes to the circulation, the mature B cell. If in another scenario, if it interacts with the self antigen, the first there could be two scenarios. One thing is either it could die, it leads to apoptosis. Either these cells are eliminated, leads to the apoptosis, the death of these kind of B lymphocytes if they are interacting with the self antigen. And another scenario will be these cells were again now replaced by the surrogate light chain and again allowed to rearrange the antibodies molecule and try to bring such combination that will now not interact with the self antigen. And if that happens, again that mature B cell will be selected for the periphery circulation, and if not, again it will undergo apoptosis process. So third step will be now, so these are happening with the antigen presented on the surface of the cells. Another cells are presenting antigens on their surface. Now if these antibodies are interacting on the surface, this antibody interacting with some molecule which are not present on the surface, some protein molecules, some antigens. So these antigens, which are as a kind of soluble antigens, and if these soluble antigen interact with the B cells, and these soluble antigens are very strong, and they lead to cross-linking of the receptor. Okay, so there is a very strong binding. It will lead to the unresponsive. It will become unresponsive to antigen, and these are called energic B cells. Okay. In another scenario also, if the similar thing is happening, but the binding or affinity of the soluble antigen with the receptor is not very strong, that it does not lead to the cross-linking, but it is interacting, so then soluble antigen interacting, then it will become a clonally ignorant cell. Okay, so that is what happens when it is become a clonally ignorant cell. So these are the tests a B cell has to pass in order to go to the circulation as a mature B cells to fight to the foreign pathogen and work for the immunity. Okay, so in the second case scenario, you have seen after the interaction is antigen is self antigen is interacting with the B cell, then there is a arrest, light chain rearrangement occurs, and then the rearrangement, and if there is a specificity, again if there is a specificity against self antigen, then it will lead to apoptosis, otherwise it will be considered a mature B cell and selected and go to the circulation or lymph node to attack the foreign antigens. So yeah. So okay, so yeah, so the maturation process, now we know, from multipotent progenitor cells to a mature B cell, the entire process we know that is happening in the bone marrow. After in the bone marrow, when the T cell matures, it leaves and the bone marrow and goes to the lymph node where it encounters the antigen. The lymph node, here you can see it is divided into paracortex and the cortex. And the paracortex will get lots of T lymphocytes, and in the cortex where the activation of this B cells will take place majorly. Okay, so um, so this is what is happening. So when the B cell is coming here the mature B cells, okay, they are interacting with the antigen. They are also interacting with the helper T cells, which is helping them to release certain kinds of cytokines, and then they help them to move to this dark zone from which is the germinal center. Okay, so here this is a germinal center that is having a dark zone and light zone, and when they interact, after interacting with the T lymphocytes, they go to the dark zone in the cortex, and there they start proliferating and and then undergoing affinity maturation, and after that affinity maturation it will become either plasma cell or memory cells. Okay, so and this is what the B cell activation process. It can be of two types as well. So it could be a thymus independent or thymus dependent process. Okay, either of these two. So thymus independent means these, when the B cells interacts with the antigen or performs its immune response, there some T lymphocytes are also involved. Okay, so if the T lymphocytes are involved in the B cell activation, it is called a thymus dependent activation. And if the B cell is not T cell is not involved in the activation of B cell, then it is called as thymus independent activations. Okay, so thymus independent activations are, for example, the there are some lipopolysaccharides, there are some bacterial DNA or some repetitive sequences, okay, being presented to the B cell in thymus independent manner. Okay, so here you can see there is a receptor on the surface of the B cell. You can see this is the IgM, the surface receptor of B cell, and along with the Ig alpha and Ig beta. So there they are interacting with some antigens, and after that, with the help of this in the cytoplasmic cell, the Ig alpha and Ig beta, there is this ITAM. Okay, this particular molecule which starts the signaling. It starts the signaling and then performs the response. Okay, here there also in the type 1 or type 2 is there in the thymus independent. So TI1 we say thymus independent type 1, thymus independent type 2. So LPS and DNA. So in the type 1, if it interacts with the lipopolysaccharides or some bacterial DNA, so then it is a type 1 kind of response. But if in the type 2 it interacts with some repetitive structures and cytokines, then it is a type 2 thymus independent response. Then thymo dependent, thymus dependent, then it is suppose an antigen is coming, this is on the surface of the B cell, the IgG is there, antigen is coming, and it is being internalized in the internalized process, then it is being presented on the surface, then the B cell is being presenting it on the surface and with the MHC2 molecules with the help of T helper cells. So this is a T follicular helper cells. Here you can see T follicular helper cell which is having T cell receptor on them with the, so they bind to the antigen presented by the MHC2 molecules, okay, as it is internalized by the B cells. So then this T cell receptor and MHC interaction takes place, followed by the CD40 interaction with the CD40 ligand present on the T cells, and then the B7 interaction with the CD28 present on the T follicular helper cells, and all of these interactions lead to the release of certain cytokines, for example IL-21, which activates the STAT3, which is a transcription factor in the B cells, and after that activation of the transcription factor, the B cells start proliferating. Okay, so the proliferation of the B cells start after all this interaction, and that is why if the T cell is involved, it is called as thymus dependent activation of the B cells, and then the proliferation in germinal center start, and on the surface of these B cell it start expressing certain receptors, and which followed by their differentiation as well in the plasma cell and the memory cell. If we'll see in the detail, so here you can see these activated B cells which are now expressing, these are called as centroblast, and they are expressing CXCR4, CXCR4, and there is this CXCL12. These CXCL12, this also tries to retain centroblast in the dark zone. Okay, so this is happening in the germinal center. Okay, this is happening in the germinal. So after that, it gets activated by the T cells. The activated B cells come to the germinal center where they start expressing this CXCR4 on its surface, and then this CXCL12, it tries to retain the centroblast in this dark zone. Okay, in the dark zone of the germinal center. Then after this, the centroblast undergo the somatic hypermutation events. Okay, so in the somatic hypermutation, they start expressing different kinds of there are certain kinds of many different kinds of mutations occur in these receptors, and due to that, the affinity of these particular B lymphocytes now changes towards the antigen, and those cells will be selected which has the highest affinity towards the foreign antigen, and that process is called as affinity maturation. So after the somatic hypermutation, these cells, they start expressing these different kinds of receptor on them. For example, from CXCR4, now they start expressing CXCR5, and then there is this particular CXCL13 which is secreted by secreted here by the FDC in the light zone, and that tries to bring these centroblast from dark zone of the germinal center to the light zone of the germinal center. Then these are called as centrocytes, and now these centrocytes are facing many different antigens, and those particular lymphocytes which are having high affinity towards the antigen will be selected, and low affinity having lymphocytes will be removed by the apoptosis. Now when the high affinity one is selected, so they interact with the this follicular dendritic cell which is present here in the light zone. These high affinity cells, they interact with this particular the antigen presented on this follicular dendritic cells with the help of um, uh, so here, so after that, it's there is another process also which is happening. So along with this interaction, they also interact with the MSC2 molecule with the T follicular helper cells, which also after interaction, it secretes IL-21, which allow them to undergo the class switching. And after the class switching, if the as the action of interleukin 21, if this they start secreting, if they start secreting IL-4, then it will become IgE. If the chemokine is interferon gamma, it becomes IgG. If there is IL-21, it becomes IgG. So these are different kinds of chemokine, whichever starts secreting, based on that the class switching takes place. So from now from IgM and IgD, they start also expressing IgE and IgG and different kinds of cells, antibodies. So now these antibodies are secreted in the circulation, and then there are these plasma cell which start secreting, and then they goes to the bone marrow to fight those antigens that are there present in there, and also in the memory B cell which goes there, and when the further attack from the pathogen takes place, then they become active. So that was about B cell activation. Other than that, we have we can know about effector T cells. And in the effector T cells, here you can see this is a when the T cell, the double positive T cells interacts with the antigen presenting cells. Based on what type of MSC they interact, they undergo the differentiation. They either become CD8 positive and CD4 positive, and there the CD4 positive cells are called as T helper cells. They are also, they have different types. So they have T follicular helper cell, we have T regulatory helper cell which has regulatory function, and then based on different kinds of interleukins. So if interleukins are interleukin 12, it becomes Th1 helper type 1, helper 2 if IL4 is secreted, and IL6 and TGF beta is in the surrounding, that it matures into Th17. Okay, so if it is a Th1 kind of helper cell, it interacts with the antigen presenting cells, and these helper cells, they do not kill, so they help in killing. They are not cytotoxic cell, but they help in killing. So they become mixed in macrophages into a cytotoxic cells which kills the pathogen. If and after the interaction, if IL3 is secreted, so once they converted into these cells, they also start expressing different kinds of marker on their surface. If it is Th1, it is CCR5, Th2 it is CCR4, Th17 it is CCR5. Then so this Th1 basically deals with the intracellular pathogen. Apart from the internalized pathogens from the macrophages, then it also helps in the monocyte production under the effect of IL3. Under the effect of TN alpha, it allows more macrophages entry into the blood. Under the entry action of CCL12, it attracts more macrophages to the site of infection. The Th2 type cells, they kills the parasites or helminths. Under the action of interleukin 13, it activates the mucosal smooth muscle and increase the motility. And in action of IL5, it activates the eosinophils, releases cytotoxic molecules. And in the effect of IL3 and IL9, it activates the mucosal mast cell, and histamine and prostaglandins are released. Th70 takes care of the extracellular pathogen. It releases IL17 to activate the epithelial cells and perform the antimicrobial protein and peptides killing. And under the when it releases IL17, it allows more neutrophil production, and when it produces CCL20, it allows more Th17 type of cells production. So these are the effector T cells. So if we'll see the questions, so multipotent progenitor cells via their FLT3 receptor tyrosine kinase, they bind to which cells? Okay, so the MPP via their FLT3, they were able to bind to the stromal cells. Okay, they were able to bind to the stromal cells. So the stromal cells is the correct option. So which chemokine helps to retain stem cells and lymphoid progenitor cells at the appropriate stromal cells in the bone marrow? So the CXCL12 was the chemokine which helped to retain the stem cell and lymphoid progenitor cells in the stromal cells and in the bone marrow. Then the pro B cell matured into the pre B cell upon interaction of what? Okay, so there were two interactions that were taking place when the pro B cell matures into the pre B cell. There is a VLA4 to VCAM1 interaction and CKIT to SCF, which is stem cell factor interaction. So both A and B is correct. So a heavy chain rearrangement occurs in the, so heavy chain rearrangement was occurring in the pro B cell. Okay. So these are the matches that we have to do. So as we have already said, so in the early pro B cell, so it starts with pro B cell, then pre B cell, and then the immature cell. So pro B cell has having two, so early and late. So in the early phase, so first the DJ rearranging for the heavy chain takes place, and then the late pro B cell. In the late pro B cell, we have the VDJ arrangement. After that, it becomes a small pre B cell, in which the BJ arrangement of the light chain takes place, and the large pre B cell, it start expressing the pre B cell receptor, and the immature B cell has the surface IgM on it. Okay, so the B cell activation occur in, so So B cell activation occurs in the lymph node. The origin and maturation both take place in the bone marrow, but the activation takes place into the lymph nodes. So the protein antigen can induce B cell activation by, so protein antigens. So B cell activation could be by thymus independent and dependent pathway. So the dependent pathway was where it was dependent on the T cells. So independent pathway were in where B cell can interact with the lipopolysaccharides, bacterial DNA, or the repetitive sequence. But these protein antigens needs to be presented to the B cell with the help of the T lymphocytes. So protein antigens can be induced by the thymus dependent process. So given are the steps of the maturation of the progenitor B cell into precursor B cell. So the correct sequence of the event is, so first it starts with the binding of this c-kit on the pro B cell, on the stem cell factor of this then ICL7 released from the stromal cell. Binding of the weekend one. Yeah, both 1 and 3 first, followed by the IL-7 release binding to the IL-7 receptor and the expression of IL-7 receptor. Yeah, then this one and then this, and then finally the maturation of pro B cell to the B cell. For examples of type 2 thymus independent antigen is, okay, so type 2 thymus independent, so bacterial flagellin lipopolysaccharide, bacterial silver polysaccharide, so thymus independents were the lipopolysaccharides. So these are the lipopolysaccharides, and these are the sequences which were thymus independent sequences. Then somatic hypermutation of heavy and light chain variable regions occurs in, okay, so hypermutation step was occurring in the dark zone, and after the hypermutation step is done, then the cell were moving to the light zone for affinity maturation. So these are the questions for today.